Welcome to the PubMed Daily Review for March 01 Stay. Today we scanned 3,722 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, an antifungal with a novel mechanism of action discovered via resistance gene-guided genome mining by Perlati B. and Harvey C.J.B. Let's break down its key contributions. This method systematically mines fungal genomes for biosynthetic gene clusters containing resistance genes homologous to essential fungal targets. It enables targeted discovery of natural products with specific antifungal activities. Up next, our second paper out of 5, treatment of follicular lymphoma with CHOP and anti-CD20 therapy, 15-year follow-up of the SWOG S0016 trial by Shadman M. and Friedberg J.W. Let's break down its key contributions. Hello everyone. Today, we're diving into a groundbreaking study that challenges a long-held belief about follicular lymphoma. For years, this cancer was thought to be incurable, with patients facing ongoing relapses. But with advances in treatment, particularly anti-CD20 therapy, researchers are asking a crucial question: can we actually achieve a cure for advanced stage FL? This paper provides some truly exciting answers. This secondary analysis comes from the SWOG S0016 trial, a multi-center, randomized study. Researchers followed 531 patients with advanced stage follicular lymphoma for an impressive median of 15.5 years. Patients were randomized to receive either R-CHOP or CHOP-RIT. The main goals were to assess 15-year progression-free and overall survival, and to use cure modeling to estimate the proportion of patients who achieved a long-term cure. This robust design allowed for a deep dive into long-term outcomes. The results are truly remarkable. The overall 15-year survival rate was an impressive 70%. Even more exciting, cure modeling estimated an overall cure rate of 42% for these patients. While the overall 15-year progression-free survival was 40%, CHOP-RIT actually showed superior PFS at 47% compared to 34% for R-CHOP. Crucially, the rate of relapse plummeted from 6.8% in the first 5 years to a mere 0.6% between 15 and 20 years, strongly suggesting that a cure is achievable for many. These findings are a game-changer for follicular lymphoma. They provide the strongest evidence yet that a cure is achievable for a significant portion of patients with advanced stage disease, challenging decades of conventional wisdom. This means we can now have more optimistic conversations with patients about their long-term prognosis. It also underscores the immense value of extended follow-up in clinical trials and will undoubtedly reshape future research directions and treatment approaches for FL. A truly exciting step forward. Up next, our third paper out of 5, a proteogenomic atlas of 1032 brain metastases identifies molecular subtypes, immune landscapes, and therapeutic vulnerabilities by Yang Zi and Zhang Ji. Let's break down its key contributions. Brain metastases are a devastating complication for cancer patients, often resistant to current therapies due to their complex molecular makeup. This urgent clinical challenge highlights a critical need for deeper understanding. Our research tackles this by asking, can a comprehensive multiomic atlas reveal distinct molecular subtypes of brain metastases and uncover their unique therapeutic vulnerabilities? This work is crucial for developing personalized and effective treatments. To address this, researchers built an unprecedented multiomic atlas. They analyzed 1,032 pan-cancer brain metastases, alongside matched primary tumors and glioblastomas, and even patient-derived organoids. Utilizing cutting-edge techniques like RNA-seq, proteomics, spatial transcriptomics, and targeted drug screening, the team aimed to comprehensively profile these tumors. This massive effort sought to identify and characterize distinct molecular subtypes and their potential therapeutic Achilles heels. The study successfully identified 4 robust molecular subtypes of brain metastases, each with unique biological programs and clinical associations. A predictive model achieved 84% accuracy on the test set, confirming subtype reliability. Crucially, one subtype, BRMS4, showed significantly worse postoperative survival, p less than 0.0017. Furthermore, patient-derived organoids revealed subtype-specific drug dependencies, like mTOR activation in BRMS3 and CDK4/6 in BRMS4, pointing to new therapeutic avenues. This groundbreaking proteogenomic atlas provides a rigorous new classification framework for brain metastases. It offers invaluable insights into subtype-specific molecular dependencies, immune landscapes, and radiobiologic signatures. These findings are pivotal, paving the way for personalized treatment strategies that target the unique vulnerabilities of each brain metastasis subtype. Ultimately, this research brings us closer to more effective therapies and improved patient outcomes. Up next, our fourth paper out of 5, the World Health Organization's Safe Childbirth Checklist on Essential Birth Practices and Perinatal Mortality, a meta-analysis by Kaplan LC and Semra KA. Let's break down its key contributions. Globally, millions of maternal and newborn deaths still occur annually, highlighting a critical need for improved quality of care during childbirth. The WHO Safe Childbirth Checklist was developed to standardize essential practices, but its effectiveness on health outcomes has been varied. This meta-analysis dives deep into the data to understand if this vital tool truly makes a difference in saving lives and improving care. Our comprehensive meta-analysis pooled data from 3 large cluster randomized trials conducted across India, Indonesia, and Pakistan. We examined over 160,000 mother-newborn pairs, comparing outcomes in facilities using the WHO Safe Childbirth Checklist against those providing standard care. Our primary focus was on stillbirth and perinatal mortality, alongside adherence to 15 key evidence-based practices, providing a robust look at the checklist's real-world impact. The findings are truly exciting. We observed a significant increase in adherence to evidence-based practices, jumping by 24 percentage points where the checklist was used. Crucially, in facilities where checklist use was observed, stillbirth rates were dramatically lower, by nearly 10 per 1,000 births. While overall mortality didn't change across the full sample, this specific reduction in stillbirths is a powerful indicator of the checklist's potential. This meta-analysis provides compelling evidence that the WHO Safe Childbirth Checklist is a powerful intervention. It not only enhances the application of critical evidence-based practices but also directly contributes to reducing stillbirths in lower middle-income settings. To maximize its life-saving potential, future efforts should focus on ensuring consistent compliance and identifying additional factors that optimize its implementation for maternal and newborn safety. Up next, our 5th paper out of 5, first-line tislelizumab plus chemotherapy for recurrent or metastatic nasopharyngeal cancer, 3-year follow-up of the Phase 3 Rationale 309 randomized clinical trial by Yang Y and Fong W. Let's break down its key contributions. Hello everyone. Today, we're diving into a crucial study that addresses a major challenge in cancer care, recurrent or metastatic nasopharyngeal cancer. This aggressive disease disproportionately affects populations in Asia, and finding effective, long-lasting treatments is incredibly important. We're exploring whether a combination therapy, featuring tislelizumab, can offer a sustained advantage for these patients. Let's find out. This was a robust, double-blind, placebo-controlled Phase 3 randomized clinical trial, Rationale 309, conducted across Asia. Researchers enrolled 263 treatment-naive adults with recurrent or metastatic NPC. Participants were randomly assigned to receive either tislelizumab or a placebo, both combined with standard gemcitabine and cisplatin chemotherapy. The primary goal was to evaluate progression-free survival, with a fantastic 3-year follow-up period providing long-term insights. The results are truly exciting. After 3 years, tislelizumab plus chemotherapy demonstrated a sustained progression-free survival benefit, extending the median PFS to 9.6 months compared to 7.4 months with placebo. Even more impressively, it showed a clinically meaningful improvement in overall survival, reaching a median of 45.3 months versus 31.8 months. While immune-mediated adverse events were more frequent with tislelizumab, the overall safety profile was acceptable, making these findings very promising for patients. These findings are a game-changer for patients with recurrent or metastatic nasopharyngeal cancer. Tislelizumab plus chemotherapy offers a powerful first-line treatment option, providing not just short-term gains but sustained, long-term survival benefits. Furthermore, the discovery that high B-cell gene expression is associated with even greater overall survival benefit opens exciting avenues for personalized medicine. This research truly advances our ability to provide better care for those battling this challenging disease.